I'm Gurna Walat. I'm a faculty here at IQB and RCSB PDB, and I'm really excited to introduce our seminar speaker for today, Professor Mike Rout. Professor Rout is the George and Ruby G. Stevens Professor and head of the Laboratory of Cellular and Structural Biology at the Rockefeller University. He received his PhD from the MRC Laboratory of Molecular Biology at the University of Cambridge under the supervision of Dr. J.V. Kilmartin on the topic of the structure and function of the spindle pole body of yeast. After his PhD in Cambridge, he moved to the Rockefeller University where he did his postdoc under Gunther Blobel, focusing on the isolation and characterization of the yeast nuclear pore complex. And in 1997, he became an assistant professor at Rockefeller and started his own lab. He's been a full professor since 2008 at Rockefeller, and he's been focusing on structural characterization of the nuclear pore complex. He's received several awards, including the Max Perutz Prize, Jane Coffin Child's Memorial Fund for Medical Research, Rita Allen Foundation Scholar, the Presidential Early Career Award for Scientists and Engineers in 2001, and the Rockefeller University Distinguished Teaching Award in 2018. His research is focused on understanding the structure, function, and interactions of the nuclear pore complex using a variety of techniques. The NPCs are molecular machines that control the passage of molecules across the nuclear membrane, regulating how DNA communicates with the rest of the cell. He and his team are interested in understanding how the complex mediates the transport and controls the organization of the nucleus. Ultimately, they hope to gain a better understanding of the roles NPCs play in gene expression, regulation, and maintenance. His lab is also collaborating on developing new technologies to map and interpret the dynamic molecular interactions in the cells. I'm really happy that he accepted our invitation to visit Rutgers and give us a tour of the Pore. Over to you, Mike.
Thank you very much indeed. Thank you very much for having me here. It really is a delight to do this. So let's. Nuclear envelope surrounds the nucleus, where all the chromatin DNA is. The sole mediators of exchange between the nucleoplasm and the cytoplasm are these little things up here, nuclear pore complexes, otherwise, as I say, known as NPCs, right there. Now, obviously, it looks small here, but as I'll show you, they're actually on a cellular scale, some of the largest macromolecular assemblies in the nuclear pore complex. And all kinds of trafficking goes back and forth across nuclear pore complexes. Small molecules like water and ions can be passively diffused quite happily, but macromolecules like proteins and RNPs have to be actively trafficked in what is known to be a highly regulated and highly selective and rapid process. All kinds of different macromolecules can exchange back and forth across the nuclear pore complex. For example, some examples are shown here. The first is that the nucleus is at the heart of the central dogma of life. DNA makes RNA, where it makes protein. That's broken by the nuclear pore complex. DNA is made into messenger RNA in the nucleus, makes messenger RNA, then gets packaged, obviously, into RNP particles, and these then get exported out through the nuclear pore complex where they can get translated in the cytoplasm. And the proteins that do that have to be remodeled as they do so and then stripped off and recycled by import into the nucleus in order to keep this cycle going. Ribosomal proteins are a great example, made in the cytoplasm, imported through nuclear pore complexes into the nucleus and the nucleolus, where they're assembled into pre-ribosomal subunits, which have to be specifically exported out through the nuclear pore complexes. And it's this trafficking that not only maintains all the architectures and structures of the nucleus, but maintains the communication between the genetic material of the cell in the nucleus and the rest of the cell and the rest of the organism. Nuclear transport, I'm just going to go briefly over what happens in the mobile phase, is that if a protein needs to get into the nucleus, what happens is it has to have a little signal on it called an NLS, or nuclear localization sequence. That can be recognized by one of a member of a family of proteins called beta carrier-ferrins. They're a form of nuclear transport factor, or NTF. These carrier-ferrins will bind to that little NLS, and when they do so, they'll actually carry it and its attached cargo through the nuclear pore complex into the nucleus, where in the nucleus, it will meet, it does so by docking to the nuclear pore, translocating through the middle and getting released. And in the nucleus, it meets a little protein called RAN, which is a member of the RAS superfamily GTPase. So it can exist in either a GTP bound form or a GDP bound form. In the nucleus, it's in the GTP bound form, and in that form, it will bind to this carrier-ferrin. When it does so, it kicks off the NLS, and that then finishes a round of import of a cargo. Cargo export is similar, except the cargo starting off in the nucleus has to have a little sequence called an NES, which is nuclear export sequence. Carrier-ferrins of a particular kind that export are bound to RAN GTP in the nucleus, and in that form, that actually stimulates them to bind an NES, and once again, cargo docks, translocates, releases. On the other side, it meets a protein in the cytoplasm called RAN-GAP, and that stimulates the RAN to hydrolyze its GTP to GDP, and in doing so, the carrier-ferrin releases the cargo, thus finishing around an export, and also putting a little puff of energy into the whole system, which, if you write all the equations down, kind of adds up to this being like a little two-stroke motor, just pushing the cycle around of import and export. Even the RAN can be, in fact, recycled by its own little transport factor, bringing it back into the nucleus, where a protein called RAN-GEF will recharge it with RAN GTP, so keep this system powered. But as you can see, the other thing is RNA export, as I told you about, is analogous. It actually doesn't use the RAN system. Instead, the RNA gets packaged on one side of the nuclear pore into the correct form with little transport factors that will actually help it thread through. Very big RNAs actually thread through. Smaller ones can go through as blocks, but the big ones thread through. The transport factors will help it out. And on the other side, there are factors which will remodel it to release those transport factors and package it ready to be handed over to the translation machinery in the cytoplasm. And I'll return to that in a little bit later, but the important point is that these factors are often ATP driven, and so they provide power for transport. But the key is, what is going on in the actual nuclear pore complex? How does the nuclear pore complex distinguish between things that are not supposed to go across and things that are, namely transport factors? How is it that transport factors can get across but other things can't? And how does it actually manage to mediate all these different, very diverse kinds of transport pathways, very different molecules of very different sizes, and yet still very cleanly distinguish between things, as I say, that are supposed to go across and aren't? But the problem's even bigger than that, because the nuclear pore doesn't just transport, it actually does a lot of work in the nucleus in particular. It's involved in things like, for example, as well as trafficking, epigenetic control of genes. Genes come up and can actually be attached to the nuclear pore, and their expression state can be regulated by it. Once those genes transcribe, it's involved in transcriptional control and packaging, specific packaging of certain kinds of mRNAs. Maturation control, particularly making sure misfolded or mistranslated, transcribed RNAs do not get out. And also, it has a heavy involvement in the control of cell division. So because the nuclear pore is at the nexus of all these different processes that are essential to the cell, we've been uncovering a huge number of different disease states that are actually associated with change states or mutations or alterations in the nuclear pore complex and its associated machinery. Some of the two biggest ones are cancers, where alterations in the transport balance can change the behavior of the cell, and viruses, which will target the nuclear pore complex and alter its composition for their own nefarious purposes. So the nuclear pore complex structure could inform a lot about how it functions in all of these different functions and how it malfunctions in disease. But the problem is its structural characterization has remained challenging due to its size, its complexity and flexibility, and that has even proven true after the revolution of cryo-electron microscopy, as I'll show you. We do most of our work in the yeast Saccharomyces, which in fact, for the purposes of nuclear transport, is a very typical eukaryote. It has differences to mammalian transport, but mainly the similarities are so much that it's a good model system to look at transport, because of course genetically and molecular biologically, it's much more amenable to manipulation. And it, like all other eukaryotes, has standard nuclear pore complexes. This is what a nuclear pore complex looks like. They're very large. They are eightfold symmetrical discs, if you look at them from the top, with a hole in the middle that the transport goes through. That hole is filled with these wiggly filaments, hundreds of these wiggly filaments projecting in from the walls of the hole. This is called the central transporter, into the middle of the central transporter. These things are about 100 nanometers across, about 100 nanometers high. If you look from the side, they're made of a series of coaxial rings, and they have structures projecting out into the cytoplasmic side and the nuclear side. They're roughly 100 megadaltons in size, and they're made of about 30 different proteins in a total of 500 or more, 550 or so copies. Importantly, as I'll come back to, these filaments are the heart of the transport machine, because these transport factors, like carioferents, they can actually bind to these filaments with their cargo. And it's that binding that allows them to get across, as we'll discuss. So many years ago now, when I was a postdoc and still had some hair, a group of labs, including ourselves, Gunther and Chuck and Ed Hurt, and then when folks in Gunther's lab, like myself, Rick and John and Susan left, carried on trying to work out what the nuclear pore was made of. And using a lot of molecular and genetic and biochemical techniques, we came up with this list of 30 proteins that actually compose the nuclear pore, which is surprisingly small when you think that a ribosome, only 4 megadaltons, has over 80 proteins making it up. But this thing, at 100 megadaltons, has only 30. But the trick, of course, is, as I showed you, it's very, very symmetrical, eightfold symmetrical. But there are other symmetries, as I'll show you, inside it. And so what happens is each of these proteins here is present in many copies, 8, 16, 32, 48 copies to make up that mass. We noticed, as well as these blue proteins that make up the structural framework, there are other proteins that actually form up this, seem to have these fluffy, disordered regions in them, extensive. And what we know about them is they seem to localize, as I showed, to the central transporter, the central hole, filling it up. What we know is they are highly repetitive and disordered. They line the transport pathway, and as I said, they are the docking sites for transport factors. This is what they look like. This is only about 4 of the 50 repeats that make up these disordered filaments. This is just one typical filament. And they're actually made of these phenylalanine glycine repeats, spaced by very, very hydrophilic linkers. So they seem to be the key to transport. And indeed, as I said, you can see that the carioferents actually dock multiple copies of these little phenylalanines, or can do, because the carioferents and other transport factors like them have little grooves running along their surface that the phenylalanines can just dip into. And they seem to dip into it very quickly. They can dip in and dip out very quickly. So we have now all the components of the nuclear pore, known as nucleoporins or NUCs. That's what we call the components of the nuclear pore. Our lab has been striving over now a ridiculously long time to attempt to fit those nucleoporins into a map of the architecture of the whole nuclear pore complex. And the precision of those maps has fortunately increased considerably from starting with a very low resolution cryo-EM map, then a what we call affectionately the blob map of being able to position where the total proteins are, all the way now to getting to near atomic resolution for much, if not all, of the nuclear pore complex, which is, as I'll show you, rather than solving everything, has just opened up some new questions. The way we have to do this then, because there's no one technique like crystallography or EM that will give us all of the answers, we have to use multiple techniques that are orthogonal that give us different kinds of information about the organization of the nuclear pore. We collect all that kind of information, morphology from EM and X-ray and SACS, stoichiometry from quantitative mass spectrometry, and connectivity using cross-linking mass spectrometry, where we use a chemical cross-linker that can link between two residues on in a complex. And of course, because you know the length of that chemical cross-linker, if you can detect what it's attached to, that tells you the distance, the maximum distance between those two residues. So it's a bit like an NMR restraint. And if you get enough of them, it can be enormously helpful in calculating the whole structure. Then with André Charly's lab, we use integrative modeling approaches, which I'm sure many of you have come across, where we take all of the data and then interpret it in terms of spatial restraints, which are essentially like three-dimensional energy fields, three-dimensional springs. And what we want to do is the spring is at its most relaxed when the data is most satisfied. So we try and let the computer relax all the springs all at once and converge to a set of, hopefully, if we've done everything right, very similar solutions that represent the architecture of the complex. Once we've done that, of course, we analyze and validate the results and then go back to do it all over again to get better and better. Of course, this has involved a huge team of people. Javi in my lab in Alona and Paola were crucial for doing the biochemistry and the isolation of nuclear pore complexes, because as I'll show you, one of the things we do is we look at single particle cryo-electron microscopy of isolated whole nuclear pores, pull the whole 100 megaton thing out, put it on a grid. Wenzhou, Yi, and Brian were crucial for the quantitative mass spec and also the cross-linking mass spectrometry. Martin worked with Brian on an amazing thing where he had a machine that could actually fly the entire nuclear pore complex on a mass spectrometer and actually measure its total weight, which gave us, the total mass of this. Jen worked by fluorescence microscopy with his group, her group, to help quantitate a number of standards in the nuclear pore to allow our quantitative mass spectrometry to be fully quantitative instead of just relative. We got absolute numbers for how many copies of each protein there were in the nuclear pore. Krishna and Steve were absolutely instrumental in the cryo-electron microscopy, single particle, and Chris I've been working with for many years. And Elizabeth and Dickvijay did some wonderful thin milling cryo-ET, so we could have what the nuclear pore looks like in the context of the cell itself, not just in isolation. John has worked with me for a long time doing functional assays, and SJ, Andre, and Ignacia, of course, were the integrative modelers. And that has allowed us, with getting these maps, to look at a lot of things, such as the functional anatomy of the transport machinery and the mechanism of transport, as well as the mechanism of RNA remodeling and export that I was talking about. Also, I showed you the chromatin, epigenetic control of chromatin happens at the pore. And these diseases that I mentioned, we can finally look at what the disease malfunctions do to the normal functionality of the nuclear pore. Today, of course, I don't have the time to go into all of that. I'm just going to function today on that tour of the nuclear pore, the functional anatomy, and if we get time, what it means for the mechanism of nuclear transport. The latest data I'm going to show you was very much augmented by some very recent data where we got new maps from the isolated nuclear pore complex, the single particle, which gave us particularly good information on the more central regions, and complemented by this thin milling cryo-ET of the whole nuclear pore, which, though lower resolution, gives you the whole thing at a reasonable resolution. And here are the two kinds of maps that we were getting, the in situ here and the isolated map here. Very much complicated with a very large new cross-linking mass spectrometry data set to give the relation of where all the residues are with respect to each other, allowing us to really calculate some new and much higher precision maps. So let's start this tour then. The overall arrangement of the nuclear pore starts, it's obviously a disk that sits in a grommet in the double membrane nuclear envelope. The membrane comes in here, goes sharply curved, and then comes out the bottom here. So there's the outer membrane, the inner membrane here. In the lumen of that EM, circling the grommet that this nuclear pore sits in is this membrane ring. And it has a bunch of little, like, niddings coming through the membrane, and they connect to this very densely packed inner ring structure in purple here. That inner ring is flanked by two outer rings in yellow here. Attached to the outer rings are some machineries on the cytoplasmic side, called the cytoplasmic export platform, and the nuclear basket on the nucleus side. And they're heavily involved in RNA processing and transport, as well as on the nuclear side, other processes as well. This shows where the attachment sites for those wiggly FG repeats are on the inside wall of the central transporter. And of course, the FGs themselves reach out and fill in that central hole and sort of project out. And just to remind you again, they are the docking sites, the transport factors, allowing transport factors to get across the nuclear pore complex. I'm going to use this little map to guide you around. So the yellow here is the outer ring, the purple is the inner ring. This is brown, the membrane ring, and this green is the central transporter region there with the FG repeats. But of course, I told you this whole thing is 8-fold symmetrical. So really, I'm showing, these would be divided into 8 spokes that just kind of continuously overlap to give that 8-fold symmetry. So what this has given us is kind of like an engineering blueprint of the nuclear pore complex. And this blueprint can be used to give us certain kinds of functional structure information about the nuclear pore. Things like, as I'll go through in order, the structure and resilience of the scaffold. Where does that come from in its architecture? How is it attached to the nuclear envelope? Are proteins in the nuclear pore complex, once it's assembled, do they just stay there like bricks in a building, or can they actually come on and off and exchange and a bit more dynamic? And lastly, what does the map tell us about the organization of the transport machinery itself? So I'm going to raise a question here, which is, how is the nuclear pore complex built analogously to a suspension bridge? And we'll come back to this again in a few slides. Starting first, though, with what we call the core scaffold, which is made of that yellow outer rings and the purple inner rings. Here it is, the core scaffold in all its glory. 8-fold symmetrical, as you can see, bunch of coaxial rings, as I said, made of the outer and the inner rings. Focusing first on the architecture of the inner rings. The integrity of the nuclear pore as a whole, it turns out, depends on a rigid diagonal column reinforcement, which is at the heart of those purple inner rings. What I'm showing diagrammatically here is 3 spokes of the inner rings, just rolled out so you can look at them. What they look like in 3D is that, the blue diagonals being flanked on either side by the outer rings, but these things are at the heart of the inner rings. These diagonal cores make many, many interactions that we'll see as we go through the rest of the slides with all the components of the rest of the nuclear pore complex, really anchoring them all together and forming, therefore, the keystone of the nuclear pore. If you mess around with this central region, the nuclear pores are very unhappy and will not assemble properly. But what's interesting is, outside of this very rigid central diagonal support column, there's more flexibility. If we go to that same diagram, but look at the joins in between these 3 spokes, they're actually quite small and they're actually very flexible. In fact, they're very hinge-like. They're even worse than hinge-like. They're actually, not only can they hinge, they can actually expand like an expansion joint on a road. So these joins between the spokes going around these rings are flexible and even expandable. The funny thing about the outer rings and inner rings is, if you look at this core scaffold, it looks bare. There's really no obvious connections between the outer and inner ring, other than they're both sitting on the membrane. So what's going on here? Well, a clue came from Ed Hertz's lab, that's done absolutely sterling work over many decades. And he found one protein in particular seemed to, whilst everyone thought it was like folded protein, it actually had quite a long little wiggly extension of it that was disordered. It was like a flexible, disordered filament. But along it, it had little ordered regions, and those ordered regions were binding to the other folded nuclear pores. So what it was doing, it was making little flexible attachment sites to different nuclear pores. That was almost an oddity until we actually solved the first full structure of the nuclear pore complex and found, to our great surprise that in fact the nuclear pore architecture and core scaffold in particular is riddled with flexible connectors that go from protein to protein. They run all the way from the cytoplasmic face here. We're looking at 3 spokes of the nuclear pore from as if we were standing inside the central transporter here. They run all the way down each spoke from the cytoplasmic to the nuclear side. And they also run, if we turn it on its side, this now is the curve, the outer membrane curving in. This is the curve of the pore membrane and going out to the inner nuclear membrane. So this is a slice through the middle of the nuclear pore from the cytoplasm to the other. You see that flexible connectors anchored at the membrane go all the way out to the wall of the central transporter. So this thing is riddled with flexible connectors. This is just a diagram of who's being connected by them, which is pretty much everyone. They run the entire length and radius of each spoke. And they provide flexible links that tie together all the major modules in the nuclear pore complex. I'm going to take a slight diversion here because, okay, now we know that the core scaffold, although its heart is quite rigid, is actually quite flexible in the joints between all the rings. And moreover, the rings themselves are connected by flexible linkers. How dynamic then is the nuclear pore in terms of its composition? As I raised earlier, do things come on and off it, or do they stay on it? So to answer the question of exchange, Jan, a graduate student in my lab, came up with a rather fancy pulse chase experiment where, I won't go into the details today, but basically it's a heavy light switch experiment where we ended up with all the old nuclear pores fully assembled being tagged and then switched it so that any new nuclear pores being assembled were not tagged. And at the switch, we changed the growth medium from a heavy to a light medium. So what happens is, if you then pull those nuclear pores out after a bit, if nothing's exchanging, the nuclear pores will remain heavy labeled. But if anything's exchanged with them, you'll see light labels starting to appear in those old heavy nuclear pores. So she did that, and we found 2 main groups, dynamics of nuclear pore proteins. The first is most of them. Most of them do not exchange at any appreciable rate. Most of them just sit there. This complements nicely work done by Martin Hetzer, who said, indeed, in us, nuclear pore proteins are the slowest exchanging among the proteins in our cells. And he looked at brains in rats and said that there were a lot of protein and hadn't exchanged over 5 years. And this has real implications for damage and aging, which we could talk about after the video. However, there is a small subset of proteins which do exchange off the nuclear pore, and they do so quite quickly. The nice thing is, instead of just having a graph, we do have a map, an engineering diagram of the whole nuclear pore. So we can heat map, color code the fast ones and the slow ones. And because it's quite a big distinction, even though this is a linear, is a gradient linear map, you see it really is yellow and blue. Yellow being the very slow exchanges and blue being the fast. Well, who are the blue guys? Well, you remember the architecture, just looking at the core scaffold, the outer ring is mainly yellow, does not do anything. The inner ring, also mainly yellow, doesn't exchange. Also the membrane ring, which is over here, is yellow, doesn't exchange. But these guys do. And these guys are all those flexible connectors. So not only then we think, are the flexible connectors able to move around in 3D, in 4D, they even come on and off occasionally to adjust themselves. Turns out that has very interesting implications for the assembly and disassembly of the nuclear pore. To get into that, I'm just going to go a bit further, and I'm going to now drill into a higher resolution picture of just one spoke unit of the inner ring, as you can see here, just like one of the 8 here. We're looking at this from the inside of the central transporter. Actually, it has 3 levels to it, and the stack levels are in this direction, but I'll show you what they look like in a second. There's the green layer here, behind it is the purple layer, and behind that is the blue layer. And oops, sorry, if you were to look at it, if we turned ourselves on the top, like this has been turned on the side, there's the green, there's the blue, there's the purple, there's the blue, and of course there's 8 of these things going around. Here is the actual EM data, just to show you we really are not pulling a fast one. You can see the helices fit nicely So I'm turning it on its side. Here's that blue layer. And that blue layer is, in fact, the diagonal strut column, okay, that we talked about. Nice and rigid. But by turning it on its side, you can see this blue layer actually has all these flexible linkers coming out of it with these little folded bits as well, a bit like nunchucks. And they go all the way through to the inner wall of the central transporter. Obviously, this would go all the way around that with a big hole in the middle. So threading from this rigid outer diagonal column comes all these flexible linkers, and they really do tie the whole thing together. If you look on the side, you can really see how they do. They not only tie together the inner ring and the adjacent spokes, they even reach out. Now, this is the inner ring. Here are the two outer rings, and you can see them reaching out to connect to the outer rings. So they really, really do riddle the whole thing with these cables. Here's a sort of wiring diagram of these cables. Don't need to go into it, but what you do see is that all of these flexible cables have these little folded bits that attach to all the proteins in the inner ring here, and I'll show you in a second, in the outer ring, tying it all together. Now, in some cells, including ours, many cells, when cells divide during mitosis, they have to take the nuclear pore complex apart. They have to disassemble it. They do so by hyperphosphorylating the key proteins in the nuclear pore. For the longest time, it's like, why are they doing those particular proteins? Well, so we mapped where the hyperphosphorylation sites were in the nuclear pore, and lo and behold, they're on the attachment sites of the cables. So this is kind of like your shoelaces. You hyperphosphorylate on the attachment sites, they come undone, just like undoing your shoelaces. When you do that, because these things are connecting all the major structural modules, the modules just fall apart. At the end of mitosis, you just reverse the phosphorylation, and they just tie themselves together again. So it really does give us an impression, and I should show you too. Here's a big, long, flexible connector going from the outer ring to the inner ring, and once again, the phosphorylation. Actually, this one is one of the most hyperphosphorylated of them all, and it's phosphorylated along the attachment sites it makes so that you can undo the inner ring from the outer ring. So you can do this reversible mitotic disassembly and assembly. Now turn to the next question. I think you can see where the resilience but flexibility might come from. Now turn to the next question is, how is it anchored? How is the nuclear pore complex anchored to the membrane? And how does it, how is the shape of the membrane and the shape of the pore defined? So we're looking at the membrane rings here, but also the outer rings too. So the attachment to the nuclear pore, this is where we are now in the orientation. This is a cross-section from the cytoplasm to the nucleoside. Here is the membrane ring. Here's the membrane attachment sites. Here's the membrane ring here. Here's the membrane attachment sites here. What we have is a major protein forming that membrane ring, that brown membrane ring going around the lumen, perinucleus externa, on the outside of this curve. And it's actually made of a whole pile of repeats of these very tough IgG folds or IG folds. We still don't know exactly how they integrate to form the ring. We're still working on that. But they clearly intertwine to form a very tough ring. From that ring come a whole pile of transmembrane attachment sites, as well as these things called membrane binding motifs, little things that will attach to one side of the membrane but don't necessarily go all the way through. So there's a big equator there. But what we also notice is, if you look on the side, there are membrane attachment sites that also go vertically all the way around. So the membrane interacting regions, as I'll show you, anchor the nuclear pore to the nuclear envelope, but they do also shape the nuclear envelope, because the equatorial ones form a girdle attaching it in the right-shaped ring around the equator of this reflex grommet. But those vertical membrane binding motifs going from the tip to tail also define the curve this way in the nuclear envelope as well. So these membrane binding motifs really help define the shape of the hole and therefore the shape of the nuclear pore that assembles it. Recently, we've got a much higher resolution picture of that membrane region, but it'll also show you that we've still got a ways to go in some of our understanding. But what we were able to resolve, this is Chris Aiken's fantastic EM work, that the membrane attachment site of that membrane ring to the rest of the inner ring involves 10 transmembrane domains from 2 proteins as a really tight bundle anchoring this thing. This thing's not messing around. And in fact, 2 beta propellers from NUT 157 make very strong interactions on this surface here. And it's analogous here. We don't have a good picture of NDC1, but we think it's a dimer. We think it's got 6 or 7 transmembrane domains. And again, this guy has beta propellers making strong interactions there. So this thing's really screwed into the membrane there. This is the shape of the ring that we see by EM. We're still trying to work out how the IG repeats fit in, but that is work in progress. But one of the things we noticed is the average diameter of nuclear pores in vivo, in situ, is around here with a 585 angstrom central channel. But we actually see a big variation in the diameters. And when you actually isolate the nuclear pore, they contract down to really close to the smallest diameters you actually see in living cells. So they're kind of relaxed. And when they do so, the diameter is smaller, 425 angstroms. And you see now this outer ring, instead of being stretched out, bows in. You can see this, the difference in size like this. So what this has made us wonder is maybe that outer ring region is involved in maybe delimiting how much the nuclear pore can expand. What we think might be going on is that when you've got a low flux, the nuclear pore tends to contract slightly, or can be contracted to reduce transport. In very high flux or very large cargoes, it can expand out. But the problem with an expansion joint and the hinge is they can go too far. So we think that this guy, this outer ring, actually prevents over dilation and the whole pore just falling apart. This actually fits quite nicely with some work from Martin back in Pomby and a bit in vertebrates too. Okay, let's move away from the sort of center of the nuclear pore and go back to those structures that are peripheral to it, namely the cytoplasmic export platform and the nuclear basket. These are the things that I told you are involved in, this guy's involved in a lot of stuff, chromatin, gene regulation, cell cycle. But one of the big things it does is it grabs the RNAs as they come in, helps to remodel them, thread them through as RNPs. And out on the other side, this guy grabs them and strips off the transport factors, remodels them again, and feeds them into the translation machinery. But today we're just going to focus on the nuclear basket. And this is recent work from our group, and particularly DBJ and Elizabeth, doing cryo-ET, which gave us some really lovely basket structures for both yeast and mouse, which I can show you. And then again, Ignacia, Neelish with Andre doing integrated modeling, and Trevor in my lab did a lot of work, as well as, of course, a call out to Kelly and Brian and Javi and a number of other people who have already mentioned helped move this forward. So here's a movie of the latest structures. Now, what you're going to see is proteins as blobs, because this is still low precision. We're getting more data to really clean it up. But this is mouse and this is yeast. And you can actually see the arrangements are similar, despite 700 million years between them. The blue is this protein NUT60, this is NUT2. They're both associated with the basket, and they make a strong connection at the foot of the main strut of the basket, helping to anchor that to this outer ring. The basket itself, the struts seem to be made of very long, disjointed segments of coil protein called TPR in mouse and NRPs. And then they form this kind of blobby distal terminal. It's a bit weird, but a lot of this mass we're still not accounting for. There's a huge amount of mass out there that we haven't accounted for. We think this is where a lot of those RNA processing sites go. And I'm going to give you a sneak peek of where we're going in the next direction. We have just got a huge data set of cross-linking mass spectrometry of the basket. And what you're seeing here is that data set and showing what kind of proteins we're finding. And as you can see, it's an absolute smorgasbord. We can see the proteasome, we know where it's docking. Ribosomes probably going through the basket, exporting mRNP assembly export factors, chromatin, cell cycle, and other things. So this is very definitely very early and very definitely work in progress. It's going to take us a long time to sort this lot out. But, you know, keep me off the streets if we can get grant money. One thing I've almost always got asked at a talk, which is the $60,000 question. Are all nuclear pores the same? Well, obviously, between different species, no, you get considerable species variation. But what about in the same organism? What about in the same cell? Well, yeast is a great system for this because it's a unicellular organism. So we ask that question. Are they all the same in the same cell? And the answer is definitely not. It's quite remarkable. We found at first two forms. One, which only has a single outer ring on the nuclear side of the nuclear pore. But there's another which has two outer rings on the nuclear side of the nuclear pore. It's even wackier. In order to join those two rings in a reticulate fashion on top of each other, they're kind of staggered, we have these densities which we're able to partially fit some proteins to the point at which we know what they are. They're actually inner ring proteins from here that have got repurposed to help hold together the two outer rings, okay? And it gets even stranger than that. So we have these two forms, the single nuclear ring, the double nuclear ring held together with purple inner ring proteins, whilst the inner ring still has all its inner rings, they're just making extras for this. These are the two forms I'm talking about, the single and the double, both of which have baskets, but there's a third form, which is single ring only, and no basket. And they all come from the same cells. In fact, we did, with Jen Gerton, who I mentioned, a bunch of, they did some beautiful, elegant experiments to quantitate proteins on nuclear pores in cells and show that indeed there are, you know, these forms do all exist in the same cell. So why? Well, that's a question for afterwards. We don't know, though we have some thoughts. But the important take home is the nuclear pore complex has evolved to have a Lego-like architecture. You can add bits on, you can put extra copies on, you can take them away, you can even swap where they are for whatever reasons that you need to change to microtune functionality of the nuclear pore. Which leads us back to that question I asked earlier, which is, how is the architecture of the nuclear pore reminiscent of a suspension bridge? Well, like a suspension bridge, it has to have a firm anchor to the substrate. And as we saw, the membrane binding motifs and the transmembrane domains form a very strong anchor girdle with, in some cases, the transmembranes have 10 transmembrane domains going through, anchoring it in, and also forming these vertical membrane binding motif lines that then help anchor it and form the curve in the other direction. It's at its core, like the struts and the segments of the road on a suspension bridge, made of very strong, rigid elements. But they are flexibly joined to each other by hinges, just like the hinges and expansion joints of a bridge between the spokes and between the structure parts. And like a suspension bridge, it's riddled through with flexible, dynamic cables that allow the thing to expand and contract and to move and to be stretched without the thing just ripping apart. Because what it has to do is define, as we see, this whole, this high density transport pathway that can tolerate a huge transport flux whilst being strong but flexible enough to manage the occasional passage of very large things, also deal with very small things. So that then brings us to the last couple of slides, which is the design of the central transporter. Am I okay for time? Yeah, I've got about 5 minutes. Started a bit late. Let's look at the central transporter. What do we know of the overall sort of design of the nuclear pore complex? The first thing is there's no motor, and in the nuclear pore complex itself, there's no energy sources, no ATPases. Those are all outside the GTPases that we're talking about, ATPases. It forms, however, a robust scaffold on the membrane to define a very clear central tube that things can go through. And that tube is absolutely filled with these wiggly filaments that also are just got loads of docking sites, hundreds, thousands of these FG docking sites. What we think is going on at the sort of 30,000 foot level is that a normal protein that cannot bind these wigglies, this is entropically very unfavorable. If this comes in here, then it has to push aside, compress the wiggly filaments. That, of course, is entropically very unfavorable. It's like pushing on a spring. Small things don't have to push much, so they can get through. But once you get above a certain size, the energy that you have in thermal diffusion in a cell is just not enough to push your way through. But if you are a transport factor that can bind these FGs, well, two things are going on. You still get the push, but you also get, if you like, a pull of being able to be transiently attracted to dozens, hundreds of these little FGs. So you can actually come in and bounce from FG to FG instead of being kicked out and actually go all the way across the nuclear pore complex. We call this virtual gating because instead of a physical gate opening, which some people thought when we proposed this a long time ago, 2000, in fact, many people thought it opened and shut. It doesn't. It only appears open if you can bind, and it appears shut if you can't. You might like to think of this in an analogy to an enzyme, which is a catalyst, but instead of a catalyst for a chemical reaction, this is a catalyst for motion from one side of the nuclear pore complex to the other. And what we say is, if you are just a random protein that can't bind the nuclear pore complex, what you experience is only the T delta S, the very unfavorable entropy of trying to get through here. That means the overall delta G of crossing the nuclear pore is too high for the thermal energy you have at the time, just for diffusion. But transport factors with their cargoes, by virtue of all these very fast interactions, you've got a T delta S just like this, but you've got a delta H of the binding offsetting it. That, of course, adds up and lowers very much the delta G of the whole process into a range where, in fact, it can happen. Thermal energy can just move you across the nuclear pore. And then, of course, you have these ATP, GTP reactions that trap the cargo on one side or the other that I showed you at the beginning. So we envision, this is the nuclear pore, these little green blobs are where the FG's repeats come out from, that it sort of looks something like this, that it's a central hole filled with these wiggly filaments like that. And they're constantly diffusing around, wiggling, and they don't like being compressed, so they have an entropic force which pushes things away unless they can bind. This is sort of just an average density zone. You can see as you come in, you're going to get a very high density of these things. But is that all it is? Well, no. We can return to our structural studies, which included, as I said, quantitative mass spectrometry. Ask what is in there and how much amounts. And this doesn't just include the nuclear pore proteins, it includes everything, including transport factors. What we've seen is that when you do EM, not only do you see the structured parts of the thing, you see this fuzzy blob, quite dense, indicating there's a great deal of mass in there, probably significantly more than accounted for by the FGs. And indeed, if you do some accounting, you find that all of nuclear porins add up to about 52 megadaltons. But there's another 26 megadaltons in our isolated nuclear pores that is accounted for by transport factors and cargoes stuck once we isolated them in this central region. So the central transporter is not just FGs. In fact, the mass of FG repeat, those wiggly filaments in the central transporter, is only about 9 megadaltons. But the mass, as you see, of transport factor cargo is 26 megadaltons. So when you see these models out there of transport, many of them do not account for the fact that only 1/4 of the mass of the central transporter at any one time is FGs. It's you know, like describing a cup of coffee and only talking about the milk. There's a lot more in there. The bulk of what's in there is transport factors and cargo, and there's also some element of organization. When you look at this blob and threshold it, you can actually see these bundles connecting. You can see them, and they come from where the FGs emanate. So these must be bundles of FGs going in and then spreading out. They must actually bundle first and then go out. And these bundles exist both in the isolated nuclear pore, but in case you think it's an artifact, they are also present very clearly in the in situ filling nuclear pores. And you can see that here too. Not as well, but you do get the same thing. There's this central plug, very dense plug, which appears to contain a lot of transport factor cargo, and then you can see these little nubbins coming out to connect to it. This is an idea that just one of these emission sites, you have a bundle here, and they spread out. And then you've got the body, which in a ring would merge into a plug-like thing with these gaps. They're really not gaps, they're lower density areas. Just to throw even more of a curveball, and probably by now totally confuse you, and I totally apologize. There's more than one flavor of these FG repeats. I won't go much into it, but they are different kinds of FG that seem to have different behaviors and definitely have different affinities for different transport factors, which raises a still unknown question, which is we think, but we don't know. That there may be these flavors that are present in different places, as you can see by the color coding in the nuclear pore complex central transporter, may actually define different routes for different kinds of transport, which could be very exciting, but we really don't know much about that. But coming back to reality for a second, last couple of slides. Is this really what you see in a dynamic nuclear pore? So to answer that, we work with Rob Lynn's lab, fantastic, and a world expert on atomic force spectroscopy, with Javi and Liz and other people in my lab and their labs. And we did atomic force microscopy on our isolated nuclear pores. And this is high-speed atomic force. Now, it's not of the same speed that you could get in a, if we were really looking at the nanosecond, millisecond scale of motion. But you can imagine how it works is it's like it's strobing, like one of those strobes on a dance floor. You can see the individual motion, but you can't see the frames in between. But what you can see is stuff is really moving around. It's a bit difficult, but you actually can see little filaments. And of course, what you see is a denser region in the middle. We know through a number of experiments that is definitely transport vacuum cargo. And it's kind of being wiggled and bounced by virtue of being on and around these wiggling filaments that are moving constantly. So clearly the central transporter is very dynamic. Now, I could bore you even more by going into a whole pile of other things, but I think maybe, to me anyway, and I could be dead wrong, the proof in the pudding is, can we actually make something that is of the same general structure of a nuclear pore, but entirely artificial, and we just add things in one by one, and do we get the same kind of appearance? So to do that, what we need is a permeability barrier in a hole based on our data. It should be supported by a cylindrical scaffold, should have grafted FG regions, the intrinsically wiggly things, right? And the distances should not be too long. They should be what we see in the nuclear pore, that is, they don't massively overlap with each other. They should not, if we look at them, be constantly coalescing. They shouldn't glue up. They should just fluctuate, wiggle. And if we added transport factor to that artificial system, we should see the central plug reappear, if that really is made of transport factors. So to ask whether we could do this, we turned to a super talented guy from Yale, Chen-Jiang Lin, incredibly lucky, super nice guy. All of these folks have been wonderful to work with. And he works with DNA origami, and he's a real master on it now. So he made these rings of exactly the same right diameter, they're even eightfold if you look, of a nuclear pore complex. And then we looked at them by atomic force microscopy. Now, this is just one of these rings on its own. And what I'm showing here with time along this axis, unfortunately I don't have movies, but this is a line through this projected in time this way. So you can see there's very little fluctuation. This thing is obviously pretty rigid. What we then did is we grafted in the FGs into it. And you can see the density actually does fill in that hole. And now you start to see spikes and troughs. That means over time, this thing is starting to wiggle quite a lot. And in fact, if you do look at the movies, it does look just like the wiggling in the nuclear pore. Well, what about the central plug? Well, we just added back transport factor. And as you see, we get a central plug. Not only that, but it's doing what we saw in the real nuclear pore. It's bouncing around, and the FGs on it are bouncing and attaching to it and coming unattached in a sort of quasi-transport reaction. So we really do see, in this case, a behavior that is incredibly reminiscent of the primary barrier mechanism in the nuclear pore complex. Structural arrangement of the FGs within a geometrically defined nanopore, enhanced in its architecture by transport factors. So we can recreate an artificial nuclear pore. So really to get down at the fine details of the transport machinery, which is really where we are now, we're returning to integrative modeling. But in this case, instead of 3D, we're now adding the fourth dimension. And this is being done by Rout, fantastic guy, who was with Andre Charlie, now has his own lab in Jerusalem. And he has taken an integrative approach. He's taken all the data we have on the atomic scale on the behavior of transport factors and these FG repeats by NMR and ITC. He's done some significant atomic modeling of how they interact and has incorporated the whole cell transport assays that we already also have. And of course, using our latest nuclear pore structures. And as this one early movie actually of this, you can see that it's happily excluding the two blue things because they don't bind. But these red guys, the transport factor and cargo, they're coming across just fine. They actually move quite quickly. And in fact, this integrative dynamic model does recapitulate a lot of the functionality of the nuclear pore. So that's where we are right now. I'll stop it there and hope it didn't go too long. I'll open for questions.
Thank you for that masterclass in storytelling and giving us the story of the nuclear pore complex. It's really, really wonderful. Let's open up for a couple questions, maybe. Okay.
Thanks for the terrific talk. I have a question about the mechanism of the biased transport in one direction.
Yeah.
To my understanding, it looked like a Brownian-Rothschild mechanism.
Yes.
Through the RG repeat and then interaction between RG repeat and cargo protein. But to have the rectify the Brownian motion, you should have some sort of the asymmetry in the system.
And you do.
So what is that?
Well, actually, yes. The asymmetry, of course, comes from the RAN reaction. That is RAN GTP and the nucleus RAN GDP in the cytoplasm. So all the energy, if you like, is on one side in the loaded RAN GTP. So that is an asymmetry because, as I showed you, how a carrier behaves in the presence of RAN GTP is dependent, A, where it finds it, and B, what kind of carrier it is. If it's an exporter and it binds RAN GTP, it will then grab its export cargo and take that. If it's an importer and it sees RAN GTP, it will dump it. So that gives that. But there is, in fact, some asymmetry in the nuclear pore. I showed you the asymmetry of the basket versus the cytoplasmic export platform. Well, it turns out major transporters, this is another complication, didn't get into. They actually, importing transport factors, have an affinity for the FGs, but they have an even higher affinity for FGs in the basket. And so what happens is it biases the diffusion. And in fact, some beautiful work from Siegfried Musser showed that if you don't have that bias, the chance of a particle getting across the nuclear envelope, across the nuclear pore, going through instead of just bouncing back is about 50-50. But with that high affinity site, it's 30-70. It's much more likely to stay on the other side. And of course, once on that side, it's being held there, waiting for a RAN to come up, but it won't go. When a RAN gets in through a series of reactions, it releases the cargo, releases from the nuclear pore, and the carrier to go back.
Thank you. That was really fantastic, stunning work. I wanted to ask you about the work from the Grillage lab, where it shows the pores being a hydrogel, a selective gel phase, which is different than the picture that you were presenting in your presentation here.
Yeah. So I mean, the thing is that that's the model that it's a selective gel phase. I mean, I can show you quickly a slide. This is this gel phase. We actually made the gels according to his instructions, and we did atomic force microscopy on them. And what we saw is they're actually basically amyloid tangles. No one had ever done this before. They haven't actually looked at these in vitro gels. So to cut a long story short, we saw a bunch of behaviors, none of which resembled what we were seeing in the nuclear pore, except the gels seemed filled like Swiss cheese with holes, basically. And the holes were lined with little leftover FGs, and they looked exactly like the free FGs we were seeing in our actual nuclear pores. So we think what's happening is, whilst the bulk gel is not a very good mimic, and it's not just does Ed Lemke's done some very careful measurement in vivo of motion of FGs and says it's not moving like a gel. We think that the reason he can get penetration and transport factors is they're going into these holes by virtue of a mechanism that we're seeing in actual nuclear pores, but not going in through the gels. And in fact, what we did is Elizabeth has been working a lot on these like condensates, because obviously there's a sort of line from condensate to gel to amyloid-like thing, and these things actually will amyloid quite quickly, which is a real problem because that should not happen. What she did was she expressed labeled versions of all the different kinds of FG in living cells. And what happens is, firstly, most of them don't form any kind of condensate or gel. They don't like to. So whatever's going on, most FGs don't do it in living systems. Some of them do a particular flavor. But what's really interesting is they form them, and if you leave them a while, they become completely aggregated. They're just amyloid. So there's got to be something stopping that happening in the nuclear pore. They form in the cytoplasm, and they form in the nucleoplasm, which means the protein is getting across the nuclear pore to get into the nucleoplasm, form these little blobs. But you never, ever, ever see one on a nuclear pore. They're going through the nuclear pore as monomers, presumably, but they don't aggregate on the nuclear pore. Something is stopping them forming a gel on the nuclear pore, which to us indicates not only is that not the state, there is some state that is preventing the formation of the gumming up of the nuclear pore into some kind of gel. I mean, it's very hard to explain how a gel-like mechanism could work for something to pass a ribosomal subunit in 4 milliseconds. You know, I mean, that's just rocketingly fast. So we do not favor that model. None of the, I mean, this is the thing. A lot of this, as I said, you can see is in vitro. The gel stuff is in vitro, making little blobs like this and showing pictures of the gel. We're just asking. We're not presupposing what the state is. We're just asking what is the state based on looking at real nuclear pores and asking how they behave. And we don't see this kind of behavior. Yeah.
So, Mike, that was a fantastic talk. Thank you. Let's see. You mentioned briefly that at mitosis, you envision a mechanism for disassembly. Can you tell us more about that?
Well, embarrassingly, not really, because yeast. The dirty little secret is yeast don't do this. The only reason I've got the chutzpah to stand up here and propose it with yeast data is because a very closely related Ascomyces aspergillus does do it. So there's nothing fundamental evolutionarily about it. If you go across the organisms, across a tree, one will do it, one will partially do it, one won't, one will, blah, blah, blah. So, but because we can't look at it in yeast, we're not done any more of it. But Jan Ellenberg with Andre Charlie and others has done some really lovely work, and they've actually modeled mitotic reassembly. It's well worth looking at that paper. But what's interesting about both mitotic and nuclear pore assembly is you'd think it would be blindingly fast, that, so as not to leave a hole in the nucleus, it just goes boom. Not so. It actually takes tens of minutes to assemble a nuclear pore, which took me by surprise. And in fact, the last component on, even in yeast, takes about a minute to fully go on, which has interesting implications if you remember that the yeast cell cycle is only like an hour and a half. But that's a whole other conversation.
Okay, in the interest of time, maybe we should leave the questions for later. But I just wanted to say many of the integrated structures that Mike showed are available on PBIHM. Many nuclear pore complex structures are in PDB as well. A lot of interesting work going on there. Please join me in thanking Mike Rout. Thank you very much for the wonderful